Good morning or good afternoon, depending on when you're watching this video. So, today we are going to talk about mitosis versus meiosis. Now, there is an assignment associated um, with this presentation. Now, you will have your choice with this assignment. You can either click the link listed below, right, and go to Flipgrid. You would use your cell phone and record a very, short or very brief video explaining in your own words the difference or a difference between mitosis and meiosis. Okay. Very straightforward, right? If you do that, you will receive 100% for today's assignment or this week's assignment. Now, if you don't want to record that video for whatever reason of you explaining the difference between mitosis and meiosis, there is another alternative to complete a little quiz. That quiz is going to be listed below as well. Okay, and you can complete that and receive your credit. It will be graded and you receive credit for the assignment. Okay, so those are your two choices. If it was me, I would do the flip grid. It's a lot easier grade. We want to see, that you guys are getting the material and being able to explain it in your own words. So I would want to, push you guys towards the flip grid video. But once again, if you guys decide not to do the flip grid video, you can answer those questions by completing that little quiz or that quiz that's listed below. Now, mitosis versus meiosis.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. How do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there, and it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So, in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So the purpose of mitosis, one purpose is to increase the size of an organism. Now, um, if you grow, if you have, you all have had growth spurts and things of that nature, you're obviously not the size you were when you were a baby, right? What's your body made up of? Your body's made up of cells. So if you get bigger, your body needs to create more cells, right? So it's used to increase the size of an organism. It's used to create, to fix damaged cells or to replace worn out cells. You know, um, obviously, like if you have a cut or a deep wound or something, right? Some of your cells are missing, right? Your cells need to end up your cells need to repair the damage or regenerate, whatever the case may be, in order to get back to normal. So they repair damaged tissue or replace old worn out cells. Um, Another thing, to reproduce identical organisms, right? So if you're reproducing, but it was just basically a copy or a clone of the original cell. Now, also, mitosis maintains a proper number of chromosomes from one generation to the next generation. So we'll just quickly go over the stages of mitosis. I'm not going to spend a whole lot of time on this. We all know about PMAT, right? So it starts with prophase, chromosomes condense. Okay, metaphase was where they align in the middle of the cell. Okay, anaphase is where they move, where they separate. Chromosomes actually separate and move towards the opposite poles of the cell. Telophase, where the chromosomes kind of relax and get ready for the cytoplasm to split and create the two daughter cells that results from mitosis. Important to remember that mitosis produces two identical daughter cells. Okay, some takeaways from mitosis. Like I just said, it creates two daughter cells. The daughter cells are 2N, so that means that each daughter cell will have the full set 46 chromosomes. Okay, um, the daughter cells will be identical, also very important to remember. And mitosis is also the process for creating somatic cells. Now, when I say somatic cells, I mean body cells, right? Now, all your body cells will have 46 chromosomes, except for your sex cells, which will have 23 chromosomes, but we will talk about that later. But the, the result of mitosis, 46 chromosomes in the identical daughter cells created from mitosis, okay? All clear on that. Now, that's mitosis. Now let's talk about meiosis.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn out cells. But not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis, you're going to divide twice and therefore do PMAT twice. And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now, I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs, and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other, and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different, though, from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line. They're in pairs in the middle. Now, during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have two newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm. But we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful of having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes and the ending cells only had 23. So they are not identical to the original and they are not identical to each other. This is going to lead to variety, a reason why two siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So meiosis is used for one purpose, really, and that's reproduction, right? And it does this through the creation of sex cells, gametes, right? What are we talking about when we talk about gametes? We talk about the sperm, we're talking about eggs, right? Egg cells. Now, as I just touched on, sperm cells have how many chromosomes? They have 23 chromosomes. Um, Egg cells as well. They don't have 46 chromosomes, they have 23 chromosomes. Okay. Now, what happens when you fuse the sperm cell and the egg cell together, right? It forms a zygote, and that zygote has 46 chromosomes. All right. So I hope that's pretty clear. So remember, we talked about all the body cells, the somatic cells have 46 chromosomes, right? The gametes, the sex cells actually have 23 each. And when you fuse them, you get your 46 chromosomes. Okay, so stages of meiosis one. All right, so let's start with prophase one. Now, these stages are similar to mitosis, but there's one huge difference, right, in prophase one. Now, when we look at prophase one, yeah, the chromosomes condense, right? But there's a huge process that goes on here that's incredibly important, right? And it's called crossing over. So this is when two homologous chromosomes come together, right? And they exchange genetic information, right? This leads to genetic variation. All right, but we'll talk about that more in a second. Then we get to metaphase, but that's a very important thing to note about prophase one. Now, metaphase one, similar to the other metaphase, right? They line up in the middle of the cell, okay? Anaphase one, they get broken apart, right? And they move to opposite poles of the cell, right? And then telophase one, you see how the cytoplasm is starting to divide and the chromosomes are starting to settle down and things of that nature. Then we move on to meiosis two, right? And here we have the same type of situation. New spindle actually forms, right? In these two cells, because remember, these are cells that broke apart or that separated, that divided, sorry. And now they're getting ready to divide again. We go to metaphase two, very similar to all the other metaphases. They line up in the middle of the cell. Anaphase, they get split, right? And they move to opposite poles of the cell. And then cytokinesis once again settle down a little bit, and the cytoplasm begins to divide, right? So have a basic cell division, which is similar, but the difference in meiosis is that we include some genetic variation, right? Because of that crossing over step that took place in prophase one. Now here, when we just talk a little bit more about crossing over, this is how it looks. You know, we have two homologous chromosomes. They come together, they align, right? To where the information is at the same point on both chromosomes, right? They cross over. So basically they trade alleles, right? Because it's the same gene in the same location. It's a different allele on the chromosome. So they kind of switch over that information, right? And then it results in these chromosomes, right, that are recombined, that that have recombinant chromatids, right? So when we talked about natural selection and things of that nature, earlier in this semester, right before before we ended up, leaving school, things of that nature. One of the big themes in natural selection was genetic variation. Well, now here's a huge source of genetic variation. It's just through crossing over and recombination, which happens when cells divide, when sex cells divide during the process of meiosis. Okay, so let's just talk a little bit more about that. It takes place during prophase 1 of meiosis. Chromosomes line up and exchange segments, and it results in increased genetic variation. Right? It's basically a mix of parental characteristics in offspring. All right. Now let's just talk about some takeaways and results from meiosis. Um, as we talked about before, meiosis. See, as we see here in meiosis one, it went to 2 cells, right? But then they divided again. So what does that mean? Meiosis resulted in 4 cells, 4 daughter cells. Okay. This process is necessary for it creates gametes, right? The sex cells, the sperm and egg. This is how they're created. Um, Basically different combination of alleles for different genes on a chromosome, right? That's really another takeaway from meiosis. And increased genetic variation. Once again, that we got that genetic variation word. It's very important for life, right? And here we see um, the source of it. Okay, so that's meiosis and that's mitosis. So now it's your turn to hop on the video and explain the difference based on this and what you've learned throughout the year about mitosis and meiosis. Explain to me the difference. Click Flipgrid below. Okay, go there, record a short video on your cell phone, upload it, and get your grade. Once again, if you can't do that for whatever reason, then the quiz is also linked below as well. All right. Very nice to see you guys. I'm looking forward to seeing your responses. All right. See you next time.